With a clear view of the primary structure of nucleic acid, and DNA in particular, let's now look at the secondary structure of DNA. The secondary structure represents the ordered arrangement of nucleic acid strands and involves two main types of intermolecular forces. We'll look at tertiary structure in a later lesson. In spontaneous forming of the DNA double helix, the two single-stranded polynucleotide strands twist around each other, as illustrated here. This model was first proposed by James Watson and Francis Crick in 1953 and was based on X-ray crystallographic results obtained from Rosalind Franklin. The specificity of interaction between the two strands is due to complementary base pairing and hydrogen bonding contacts. Note that in order to form and maintain these hydrogen bonds, the two strands adopt an alignment that is anti-parallel in nature. In other words, the 5' end of one strand is in contact with the 3' end of the other strand. By this alignment, the bases of the nucleotides within the strand are positioned to form optimum hydrogen bonding contacts. In this model, A pairs with T to form 2 hydrogen bonds, and G pairs with C to form 3 hydrogen bonds, as shown here. These are referred to as canonical or Watson-Crick base pairs. Note that this arrangement always pairs a purine with a pyrimidine, providing a consistent distance from backbone to backbone in the secondary structure. It also means that the number of purine residues will equal the number of pyrimidine residues, referred to as Chargaff's rule. Note that this rule only applies to double-stranded DNA. Within the DNA double helix, we find that the spaces in the backbone are not uniform throughout. Instead, there are two spaces or grooves of different size. The major groove contains a larger space, and the minor groove a smaller one. We will see in our next lesson that this arrangement provides optimum stability for the helix, and we'll see later that it also provides more ready access for DNA binding protein.